And this week we're doing Unit 2, which is tissues, skin, which is our first system, and then we do our second system, which is the skeletal system. And with the skeletal system, we are going to cover 3 chapters, and that's a lot. Now, when you do the bones, when do you do bones in lab? Have you looked ahead? This week. What are you doing? Today you have CAP. What's tomorrow? You have a quiz on tissues and then the first of the 2 bone labs. So we do 2 days in lab. We do 2 labs on bones. And then Thursday is the makeup CAP from last week. Okay, so you do just the 2 labs on bones this week. Now, when I get to that in lecture, I know what you do in lab. So I put it on the lecture test. In lab, you get tested over, can you name the bone, can you name the parts of the bone, can you tell whether it's a left bone or a right bone, just on the ones we assign. In lecture, I expect you to be able to associate the name of the bone with its parts, okay? So if you're studying that for lab, then you're going to get 2 grades for the price of 1, because it's also going to show up on your lecture test. And I have, in Canvas, I have study materials for you. Are you using the Quizlets for lab? Are they good? Are they helpful? Yeah. So if you use those for bones, every time you do those, you're helping to study for lecture. The one thing I don't do a lot of in lecture, because I know you do it in lab, is visual identification. So in lab, you're going to look at the real bone, not a photo of it, the real thing, and identify the parts. In lecture, I'm not going to show you bones, photos of bones, pictures of bones, because that is definitely a lab-related thing. But if you can associate the name of the bone with its parts, boom, that's good. That's going to help you on test 2. There are, though, 2 other chapters on the skeletal system. We'll be hitting the physiology of bone in here, the histology of bone in here, and, you know, let me backtrack. I think I scooted the last chapter to next week, the one on articulations, the joints. I think we wait until next week and do that with muscles, pretty sure. All right, so for today, we're going to do tissues. You did a lab on tissues. I'm going to try to relate what you learned in lab with that whole idea of complementarity of form and function, if you know how it's built, because you learned it in lab. Now we'll look at how it works. Tomorrow we do the integumentary system, which, by the way, is also the main subject of your online lab. Do y'all remember that you have a completely online lab? Does anybody remember that? Do you remember when you registered for the course and you had to register for labs that meet on Friday? And then it said, oh, we're not really meeting on Friday? Is that ringing a bell? Am I hallucinating this, or do y'all remember that? Anybody? Yeah. That was necessary to meet the contact hours required for the course, to schedule a lab on Friday, but we don't have a structure for you to be here on Friday. We have an online lab. Part 2 has been posted. It's up there. I wrote it last week. So it has to do with tissues and skin, and you should go do that. Yeah. You have no idea what I'm talking about the online lab? None whatsoever? Well, I would show you, but I got locked out of the desktop computer again, so I can't. When you go into Canvas for Lab, you will find a module that is called Online Lab. Did you do the first lab assignment, the introduction, the orientation lab? Go back and read that stuff again, because it tells you in there. Okay, it does say in there, there's an online lab. It's 5 parts. The first part was the orientation, the lab policies, and a little bit of orientation to anatomical terms. And you had that online quiz that was timed. Yeah? That was part 1 of our online lab. Part 2 is posted. It's in Canvas for Lab. Go look in Canvas for Lab and you will find it. Go look through the modules and you will find it. Okay?
It says online lab?
It either says online lab or integration lab. It probably says integration lab for y'all.
There's a biology 24 online integration lab.
Yep.
And inside the orientation.
Yeah. So there's information about it posted in Canvas, and the lab, I'm having to build it as we go. Y'all are the first ones to get to do this. So I'm telling you, the part on tissues and skin is written and posted for you. It's in soft chalk. Now, y'all are used to my soft chalk stuff by now, although I saw a couple of you haven't even done one of them yet. Do them. In soft chalk, some of you are reporting technical problems, like drag and drops won't work or whatever. All I can tell you at this point is you'll close the windows and try again, try a different browser, try it at a different time, whatever. The integration lab.
There's nothing in it. I mean, it just has a little.
But there's another, look further down in the modules, you'll find something else. Unless I forgot to publish it, which it wouldn't surprise me. But I will check again when the class is over. It's going to be built in parts. Part 2 is the part I just finished building. I thought I sent an email out to everybody in lab. Okay. That was Friday. I've slept a couple of times since then, so who knows what I did. But I'll make sure after class is over, I'll make sure it's up and running and available to you. And you should do that this week because it has to do with skin. And that's the subject of this week's information. It has to do with tissues and skin. Next week, the third part will be on surface anatomy. And surface anatomy includes things like what's that bony projection there, what muscle is this right here, to know the names of the bony parts and the muscles that you can actually observe by seeing a whole person with their skin still on. Does that make sense to everybody? That's called surface anatomy, very valuable information. And then the last part of the integration lab will be on sensory functions, which we won't do until after you've done the senses in lab. By the end of the term, you have to have that done. There's no specific grade associated with this. You already have your grade for the integration lab. It was the orientation quiz. That's the only separate grade you get for that. But the information that's covered in this integration lab is fair game for your first and second lab practicals. Okay? So what's in the parts that I've posted now and the part on surface anatomy of bones will be on your first practical. And the part on surface anatomy and muscles and the stuff on sensory information will be on your second practical. And I'm making sure the lab instructors have access to that so they know what to ask you on those practicals. All right? So are we reasonably clear? I'm clear that you're telling me you can't see it yet, and you're clear that I'm telling you to go look for it after class. Is that good enough for now? Okay. All right, so today we're doing the introduction to tissues, and in fact, you've already been introduced to tissues, haven't you? Because you did them in lab, but there are aspects of tissues that you have not yet done, and we're going to start with, I got a speaker, so we'll see if that works.
Welcome.
See if that's loud enough for you.
Waiting to connect to Bluetooth device.
I don't want a Bluetooth. Did I bring my other thing? So we're going to watch a little video on embryonic origin. How many of you, don't lie this time. Usually I do expect you to lie to me, but not today. How many of you actually did the assignment I gave you, which was to look at the germ layers? Ectoderm, endoderm, mesoderm? Sort of, kind of, a little bit? Well, that's about what I expected. So actually, we're going to go back over that. All right, so pay attention to this very brief film. It's very brief. It's like 2 minutes.
Look at the beginning of development, looking into the ovary at an unfertilized egg. It then gets fertilized by sperm, which travel down the fallopian tube. So here, millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus. And then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring, and this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life, that takes days. about 5 days. At this stage then, I'd like to draw your attention to the inside of the blastocyst where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal. And the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc, and then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo. Those lines represent sites where cells are migrating in and out. And here's an important stage when the 3 beginning layers of the embryo, the so-called germ layers, are formed, and I'll come back to that in a few minutes. As development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns, and appendages start to bud out. You see the head forming and the eye. And then eventually we get a small embryo, and some months later, of course, this would be born as a young baby.
Okay, so does anybody have any questions about the 3 germ layers at this point? We're going to watch another video in just a second. So we had ectoderm, mesoderm, and endoderm, right? Which one's on the outside? Ectoderm on the outside. The endoderm forms the inside. You know you have a tube within a tube body plan, right? Your digestive tract forms a tube inside of the tube that is your skin. And that inner tube is lined with endoderm. Make sense so far? Then what's the mesoderm? Pretty much everything in between. Okay. So let's watch a second video. This one is on differentiation and cell fate. Do y'all remember what differentiation is? That's that specialization of cells as they mature into their final form.
Move quickly the early stages of development again, and then we're going to get to this stage when the germ layers are set aside. The 3 germ layers, the ectoderm, mesoderm, and endoderm. So here you'll see then that the inner cell mass gives rise to the entire adult animal, in this case a human. This same process would occur in a mouse, as we'll see a bit later. If we now look at the embryo after the 3 germ layers have formed, this video will highlight what comes from the blue germ layer, the ectoderm. You'll see that it makes the nervous system, including the brain, and the skin. The middle part, the mesoderm, green here, gives rise to the muscle, including the kidneys, the heart, and the endoderm gives rise to the whole gut tube. There you see the lung, the liver, the intestine. And to give one more detailed example of this, let's think about the development of the endoderm, and in this case the formation of the pancreas. So there's the pancreatic bud, which comes out of the endodermal derivative. Now I'm going to talk to you a bit today about the pancreas as one example of how an organ and then the cell types within that organ get made.
Okay, and that's all fascinating, but we're not watching that part of the video. So if you want more information than that, you'll have to go back to the HHMI website and watch the rest of that, which had a lot to do with diabetes and very interesting information. But we're just at the point here where we are looking at just the basic derivation of the different tissue types so that you have some connection between how they formed in the embryo and what they do in an adult organism. There's a link, an important link there that we want to pay attention to. So once again, you see that if we start with the very beginning of things, in the embryonic development, we have this stage at which your embryo is just this little oval disc, as you saw in the video, and that oval disc has these 3 layers. And the formation of the 3 layers then is kind of a programmed set of steps, but you have your ectoderm, mesoderm, and endoderm forming. You do need to know what comes from each layer. You need to know it in the detail that is posted in Canvas for you. So just as an example of things you don't have to know, different endocrine organs are derived from different tissue types, and I've never felt the need to burden you with knowing which ones come from which layer. So there's some big picture ideas that are, it's pretty clear that you should learn, and then there's some little details that we're not going to worry about. So big picture, what are the two big things that come from ectoderm? Nervous system, your entire nervous system, and when you say skin, I know that's what he said, but it's really just the outer layer of the skin. The skin has two layers, the epidermis and dermis, and it's just that outer epidermis that comes from ectoderm. Does it make sense to you that the outside of the body comes from the outer layer of the embryo? Does it make sense to you that the nervous system comes from that same layer? Does it make sense? Is it logical, rational? You go, oh yeah, well, I would have predicted that. It's not on the outside, is it? Not really. So it connects to the outside, so you have sensory nerve endings in your superficial tissues. So from that aspect, it makes sense, but does it make sense that the brain comes from the surface? Not really, not to me, not really to most people. So, so that's just kind of weird, and there are those weird things that you have to remember. All right, so we have our germ layers, and this is a cross-sectional view, again, at a fairly early stage of development. And what I want to point out to you here is that we have the body itself. You can see where the ectoderm is here, right? Is that the outermost layer you see in this picture? Do you see another layer here? Okay, so what's that? It's labeled for your convenience. Label's way over here. That's the amniotic sac. All right, so we're looking at a stage of pregnancy here, and you just need to be familiar with that concept of get your benchmarks when you look at a picture and make sure what you think you see is what's actually there. Can you see the gut tube is colored yellow? Endoderm is yellow in this particular image. Ectoderm is what color? Look at the picture. Sort of a blue, light blue. Okay, so what's all the pink? That's all mesoderm, including the pink on the amnion. Mesoderm actually migrates out from the embryo and coats the amnion as well. So you have lots of, we call it extra embryonic mesoderm. All right, that's probably more information than we need, but it helps you to get your bearings, okay? So the endoderm at this point is just lining the gut tube. But there are things that grow off of your gut tube that you need to know of. Your liver, actually. Your pancreas, as he mentioned in the short video we watched. Even your lungs grow off of the gut tube. So they're all made of what kind of tissue? That would be endoderm. Some of your endocrine glands grow off of the gut tube. What kind of tissue are they? Endoderm. Some of your endocrine glands grow off the nervous system. So what kind of tissue would they be? That would be ectoderm. Okay, does that make sense to everybody? Okay, so that's enough about the arrangement of the germ layer. And obviously we'll come back and finish embryonic development when we get to A&P2. On our tissue classification, did you learn the 4 kinds of tissues in lab? What did you learn about muscle and nerve in lab? Probably next to nothing, right? Did you have to remember that they were 2 of the 4 basic kinds of tissues, and that's pretty much it. We'll go into a little more detail now. We're going to hold off on the specifics until we get to those body systems. Hey, we have a whole chapter just on muscle tissue and a whole chapter just on nerve tissue. So for now, we're just going to pick up some big ideas and leave the rest till later. Okay? All right, so I have a question posted at the end of the screen. Do all body tissues fall clearly into one or another of the 4 basic classes? If you were a histologist and you just looked at a section through tissue, would you be able to point with clarity? This is epithelium, this is connective, this is muscle, this is nerve. 100% of the time? Sorry, there are some parts of the body that didn't read the book and didn't know they were supposed to be classified one way or another. So you have cells in some places that are called myoepithelial cells. What would they be? What does MYO tell you? Muscle. So there are cells that are epithelial because of their location and appearance. They're clearly epithelial, and yet they contract. What are you going to do? Okay? So what a histologist does is look at their embryonic origin, and then from that try to derive what classification they want to put them in, or look at their ultrastructure, that is their microscopic structure, and say, well, it looks more like epithelium than it does nerve, but still, you don't want to be too cut and dried about where things need to be classified. Likewise, there are some of the connective tissues that are not clearly classifiable. So not all tissues in the body fall neatly into one of these classes, but most of them do. And we're not going to get bogged down in the exceptions. I just want to make sure you know that not everything is quite that simple. Okay, so here's your master list of derivation. This looks like at least one test question to me, doesn't it to you? Yeah. So notice that epithelium is derived from all three germ layers. Connective tissue and muscle both come from mesoderm. There are some exceptions to that. And nerve tissue all comes from ectoderm. So that's a list worth jotting down. By the way, I think I already posted the outline of this. I gave you the whole handout version of this PowerPoint. It's posted in the module for today's lecture. Everybody got it? Okay, so with that list then, here are a couple things I want you to think about, and this will be the beginning of what you're going to turn in. So each table needs a sheet of paper. Please put the date on it, your table number on it, first and last names, before you're going to turn around and work. And you're going to answer these questions in terms of embryonic origin, both of them. And I'm going to give you about a minute to discuss the answer. These to me are pretty straightforward. So if you don't have an answer written down yet, please start writing. By the way, occasionally I have students who don't write down any answers until I give you the answer in class. That's not the answer I want you to turn in. I want you to answer in your own words what you think the right answer is. That allows me to assess how well you're doing. So if all of you always turn in correct answers, then that means I get to speed up. So it's better to be honest and give me the answer you think and let me assess how fast or slow I need to go in class. Anybody stumped and just doesn't know what to do? Everybody's got some sense of where we are? Okay, so tell me about the first one. What's the connection between the skin and the nervous system?
They derive from the same layer.
They're derived from what layer? Both derived from ectoderm. So if you had some gene that was involved in the development of cells in ectoderm and it affected ectoderm only and not mesoderm or endoderm, then it shouldn't surprise you to see signs of it in both the epidermis of the skin and the central nervous system. Does that make sense to everybody? There is, in fact, a type of brain tumor that can be diagnosed, well, not diagnosed, but correlated with a particular kind of birth defect. Well, not so much a birth defect as a birthmark. I wouldn't call a birthmark a defect, would you? Technically, I suppose you could say it is, and it depends on how big it is and so forth. But there are some birthmarks that we specifically call the café au lait birthmarks. Have you heard of those? So what is café au lait? Has nothing to do with skin. What is café au lait? What does it mean, café au lait? Coffee with cream. That's what it means. So what's the color of coffee with cream in it? That's the color of these birthmarks. So a café au lait birthmark can be associated with a particular kind of tumor of the nervous system. That doesn't mean you should freak out if you have café au lait birthmarks. But in families where there's a tendency to have this kind of tumor, then you can often, just sort of as a first step to diagnose, if you have the birthmarks, you probably ought to be watched or assessed. If you don't, you probably have nothing to worry about. Does that make sense to everybody? Okay, second question. What's the embryonic origin of mesoderm have to do with muscles, bones, and blood vessels? They're all derived from mesoderm, right? Okay, so if you had a defected mesoderm, what might you see in muscles, bones, and blood vessels? If it's a developmental delay or a stunting of growth, if you see it in the bones, you might see it in the muscles as well. And then you shouldn't be surprised to see cardiovascular issues. Heart and blood vessels also come from mesoderm. So just as an example, in kids born with Down syndrome, in addition, I mean, they have defects across the spectrum. All cells are included, right? They have differing degrees of developmental delay. That's because of nervous system defects. But they also have characteristic physical appearance. That's another ectodermal effect. But they also tend to have heart problems and poor muscle tone and short stature. And all of those are defects of mesoderm. Make sense to everybody? Any questions about the relationship with embryonic origin and the 3 germ layers with the different tissue types? All right, so I asked you specifically, did it make sense to you that the brain comes from ectoderm? And I finally got you to admit, no, that doesn't make any sense at all. So if you look at the diagrams here, you can see that the neural tissue actually does sink in. It starts on the surface and it literally sinks in. You're looking at cross-sectional views, top right and both bottom views. And so what you have is the tissue builds up in these 2 ridges, and that creates sort of a groove in between running down the midline on the dorsal surface of the embryo. And so you get this neural groove that develops into a neural tube. So if you look up here, I'll try to sort of demonstrate that, although it's not that simple. So this sheet of paper represents the ectoderm, and you've got mesoderm and endoderm underneath doing their thing. Eventually, that whole body's going to roll into a tube, right? With endoderm on the inside forming the gut. But let's focus just on the ectoderm. What's going to happen along the midline is that it's going to invaginate like that. Okay? So it forms a groove. Now, it's not forming a groove by literally folding per se. It's actually growing more cells to do that. Very hard to demonstrate. And then eventually that groove will seal up and literally pinch off so that I'll form a neural tube, hollow neural tube, and then the skin will grow over it. Now do you see how it's going to look? There are conditions in which this is incomplete. And it may be incomplete, if this is the head end, it may be incomplete from, say, thoracic region on down, or it may just have one little spot. where it doesn't fuse, and that's spina bifida. Okay, in spina bifida, a failure of that neural groove to completely close up into the neural tube keeps it open. And because the skin doesn't grow shut over it, as the fluid of the cerebrospinal fluid starts to accumulate, it actually can push the membranes out, and you just get a bubble of membrane out the skin. And because of the effect of that on the growth of the neural tube, sometimes the neural tube defects prevent the spinal cord from fully developing. So depending on where along your back that might occur, the higher up, the worse it is. So you can have someone with perfectly normal brain development and then spina bifida, say, in the lumbar region, and then they don't have use of their lower limbs at all. Does anybody know what we finally linked to spina bifida? It was one of the most astounding moments in the history of nutrition research when they were finally able to correlate spina bifida incidence with a deficiency of a specific vitamin, folate or folic acid, which is one of the B vitamins. And when they nailed that down, I was teaching here. I was actually teaching in this room when I announced it to my students. And within weeks, I saw posters popping up in every ladies' room that I visited, particularly the gym, where they just had big posters saying, you know, take your folic acid. If you're pregnant or planning on becoming pregnant, make sure you get enough folic acid in your diet. Folic acid is not hard to come by. But in this culture, young women seem to feel like they need to deprive themselves of proper caloric intake. And so there were frank nutritional deficiencies that were correlated with that. So the incidence of spina bifida just plummeted after they figured that out and figured out a way to reach women to treat it. So that's the significance of neural tissue development. The ectoderm literally sinks in and forms the brain and spinal cord. All right, so enough about embryonic development. We're going to focus our attention mostly on epithelium and connective tissue. Like I said, we're going to do muscle tissue when we get to the muscular system and nerve tissue when we get to the nervous system. So epithelium and most of the connective tissues today, and even some of those we're going to defer until later. So epithelium, properties of epithelium are important for you to keep in mind. If you have a free surface, chances are it's coated with an epithelial tissue. Now, you need to know what I mean by free surface. So can you give me an example of a free surface within the body? A free surface. The brain as a free surface? Okay, so clearly we need some definitions of free surfaces. So if we go back to this cross-sectional view that I had, that's as good a one as any. Where is there a free surface on this picture? Anywhere there's a cavity, the tissue that lines the cavity is at a free surface, okay? So your ventral body cavity is lined by tissue. It's a cavity, so what kind of tissue lines it? Epithelial tissue. Your gut is a cavity. So what kind of tissue lines your gut? Epithelium, okay? Even your skin is considered a free surface, right? So anytime you have a hollow interior, and you should have learned in lab a term for that. The term was lumen. You remember that? Okay. So if you have a lumen, that's a hollow area. Fluid might flow through it, or it might be an air-filled space, whatever. But you're going to have epithelium lining it. There's only one place that I know of where that's not true. I'm going to go ahead and give you the exception now because we'll cover it later in this chapter, and that is your joint cavity. So some of your joints, like your knee joint, your hip joint, shoulder joint, some of the larger joints have a cavity, an opening, and a space inside. What kind of tissue would you predict lines that cavity? You're going to predict epithelium because that's kind of our definition of epithelium. You've got a free surface, you expect epithelium to get there. And in that particular case, it's not epithelium. It is connective tissue, but you know what? It looks like epithelium. So do you remember what I said earlier? You can't classify everything that easily. So joint cavities are lined by tissue that is derived from connective tissue, but takes on the appearance of epithelium. Okay? So what are the characteristics that a surface tissue needs to have? If it's going to do its job, don't the cells need to be tightly connected to each other? What would happen if they weren't? If you have a gaping wound in your skin, what does that mean? Germs can get in and body fluids can leak out. So does it make sense to you that in epithelial tissue, all the cells are tightly connected to each other? And what about in our synovial membrane, where it's not epithelium, but it looks like it? You guessed it, all the cells tightly connected as if they were epithelium. Okay, so no gaps. It also needs to stay firmly attached to whatever tissue is anchoring it. What would happen if your epidermis weren't firmly anchored to your dermis? It would just peel off very easily. There's actually a disorder, a genetic disorder, where that happens. And the skin just develops big blisters at the least provocation, least pressure at all, and just peels loose. And that's not good. So we appreciate that our epidermis is tightly connected to the underlying tissue, and the same is true of all of our other epithelial tissues. It regenerates quickly. Why is that important? Isn't the surface the most subject to external threats of any kind, whether it's germs or abrasion or x-rays or whatever? Everything that is going to get in to harm you has to go through an epithelial layer to do it. So which tissue is the most exposed to any external threats? That would be the epithelium. So it makes sense, doesn't it, that it regenerates quickly. Lifespan of an average epithelial cell that is on the surface of the body, 3 to 4 weeks. That's a pretty short lifespan, don't you think? What's the lifespan of your red blood cells? Anybody know? Red blood cells? About 100 to 120 days. 120 days is what, 4 months? And those cells don't even have a nucleus, and they last 4 months. So how about your epithelial cells in comparison? 3 to 4 weeks. So very short lifespan. Regenerate quickly. Covers all the blood vessels. Does that not seem obvious to you? So do you have blood vessels in your epithelial tissues? Are they inside of the epithelial tissue itself? Nope, they're under it. They're deep to it. Okay, so we have a term for that. We say that epithelial tissue is avascular. What does that mean? Avascular. Whenever you put a at the beginning of a word, it means absent or without. So avascular means epithelium doesn't have any blood vessels in it. There are blood vessels up just right up under it, but not in it. Okay, and I can possibly demonstrate that that's true. You know your epidermis is an epithelium, right? Epidermis is the outer layer of the skin. Have you ever got a paper cut that hurt but didn't bleed? Yeah, makes you want to say bad things. Yeah, why doesn't it bleed? It didn't go deep enough to get all the way through the epidermis. Okay, are there sensory nerve endings in your epidermis? Oh yeah, that paper cut hurt, but it didn't bleed because you didn't go all the way through. Make sense to everybody? Questions about that? And then the last thing on our list, we do park nerve endings, receptors in the epidermal layers. In the skin, absolutely. We're actually going to be learning those, some of them now and some of them when we get to the sensory nervous system. But in your mouth and throat, you know you have thermal receptors, right? When you swallow something cold, when do you stop feeling that it was cold? How far down have you swallowed it? Or hot, likewise. Maybe if it was really hot or really cold, maybe all the way to the stomach, but otherwise about 2/3 of the way down the esophagus. And then you've switched over to the subconscious branch of your nervous system. So you do have some sensory receptors that are definitely in the epidermal tissues. All right, so those are some properties of epithelium that make sense knowing that it's found on free surfaces. Here is the diagram out of your textbook. This is one of the diagrams. I like this one because it puts the epithelium in context with the connective tissue. Which one is thicker? Definitely the connective tissue is thicker than the epithelium. That particular layer of epithelium, well, you've already been to lab, right? So you can classify that one for me, right? What is that one? Is it simple or stratified? It's a simple. What's the shape? Columnar. Okay, so that's a simple columnar epithelium. Do you see the basement membrane? The basement membrane is characteristic of almost all epithelial tissue. Almost all. Epithelial tissue has a basement membrane. That basement membrane is what anchors the epithelium to the connective tissue. And the basement membrane has two layers to it. You can kind of see the two layers in the picture, even though they're not labeled here. One layer grows out of the epithelial cells. One layer grows up out of the connective tissue, and it's fibrous proteins that just kind of tangle with each other, knit themselves together, and make sure it's not easy to pull them apart. Okay? So just as a check on your terminology, is the basement membrane on the basal surface or the apical surface? Basal. What is the apical surface? It's the free surface, the one at the top, at the apex or tip of the cell. Okay, so make sure you keep those terms straight. We also have a lateral surface, and depending on the kind of epithelium, that may be big or small, but the cells interact with each other laterally as well, as we'll see. All right, so that's just sort of our overview of the way the epithelium is in relation to the connective tissue. And can you tell me, why does epithelium need to be highly regenerative? Can you just write a short answer? Just talk amongst yourselves. Just write a real quick answer to that. Why does epithelium need to be highly regenerative? Okay, so who's got a good answer for that? Who has an answer for that?
Has the most exposure.
Most exposure to what?
To damage.
To damage, external threats, germs, abrasion, whatever. So that one I think is pretty obvious. I hope you all got that without too much difficulty. Okay? All right, so now we're going to focus on one of our themes, complementarity of structure and function. What did it mean to be complementary? They work together. If you know the one, you can predict the other. So the complementarity of structure and function, in the case of epithelium, has to do with, clearly, how thick the different types of epithelium are. And we have two different kinds of functions of epithelia that are in direct conflict with each other. If you're good at one, you're not going to be good at the other. Protection and transport. If you want as protective a layer as possible, is it going to be thick or thin? Protection, thick. If you want fast transport, if you want stuff to get through that layer fairly quickly, do you want thick or thin? There you want thin. Does everybody see just immediately the conflict between our two competing needs? So protection, depending on where you are in the body, you may need a lot of protection or a little bit. Do you need a lot of protection in your mouth, in the lining of your mouth? Hey, you got sharp objects in there, don't you? Not just teeth, but sometimes what you eat is sharp. Okay, so there you want a fairly thick layer. The epidermis is as thick as it gets. Okay, so how thick does it need to be? That's going to determine going to be determined by the protective needs. Make sense? On the other hand, for transport, if the main job of an epithelial tissue is either to absorb stuff in or secrete stuff out, then the thinner it is, the faster it can do its job. So depending on where you are, how much you need to absorb, how fast it needs to be absorbed, how much of it needs to be absorbed, and how protective you need it to be. Okay? So best example of that, I suppose, is in the air sacs of your lungs. What do you do in there? Gas exchange, oxygen in, CO2 out. How much abrasion are you worried about? Are those little oxygen molecules sharp? No, that's not an issue. And you've protected them, right? You have a ciliated epithelium on the way down to remove any particles. germs are filtered out, so forth and so on. So by the time it gets to the air sacs, there's pretty much no harmful stuff left unless it was a gas you inhaled. So how thick does it need to be? It's the thinnest you can get. What's the thinnest one you can get? The thinnest kind you learned in lab. Simple. Squamous epithelium is the thinnest. What are your air sacs made out of? Simple squamous epithelium. Okay? Make sense to everybody? On the other hand, what about the lining of your stomach? Or your small intestine? Either way, it's the same stuff. Do you need to absorb stuff? Yeah. Do you need some protection, though? Yes, because you swallow germs that could still be infected by the time they get to the stomach or small intestine. So do we put simple squamous there? Let's don't. It's a little too thin to meet our needs. Okay? So we're going to go through our different kinds and kind of go through for each one on a case-by-case basis what we expect, and that then should help you make sense of why the locations we assigned you to learn in lab make sense and are easier to remember. Before we do, we're going to talk about the cell junctions, and I don't remember in your book whether this is in the chapter on cells or the chapter on tissues. Was it in the cell chapter? Okay, well, it makes more sense to me to put it here because not all kinds of cells have these junctions, but all of the kinds of junctions are found in epithelial tissues, and that makes it a whole lot easier to talk about it in context with epithelial. So we have three kinds of intercellular junctions. What does inter mean? I-N-T-E-R means between. Between. So intercellular junctions are found where? Between two adjacent cells. So we have tight junctions, desmosomes, and gap junctions. These all have other names. These names are my favorites. They're easy. So if it's a tight junction, its job is to stop fluid from leaking across. You should think of tight junctions as being like Ziploc seals. When the cells are zipped to each other, nothing leaks in and nothing leaks out. Those tight junctions are based on specific adhering proteins. You remember we talked about CAMs, cell adhesion molecules? That was on your list of cell membrane proteins to learn. So almost all of these are going to involve some kind of CAM or another. Protein locks to protein, the cells are stuck to each other, nothing leaks through. Okay? Desmosomes are like little fibrous stitches that just kind of sew the cells to each other and prevent them from ripping apart from each other. You have lots of desmosomes in your skin cells. Why? Do you want your skin peeling off? Let's not let it all peel off whole cloth at the same time. Guess what gets ruined when you get a really bad sunburn? Those desmosomes. So what does the skin do next? It starts peeling. Okay? Desmosomes prevent that. They stitch the cells to each other, and they prevent the cells from peeling away from each other. You also have lots of desmosomes connecting your heart muscle cells to each other. This should provide you with some comfort. If your heart starts contracting really strongly, is there any chance the cells are going to fly apart from each other? No, your desmosomes are going to help prevent that. Okay, stitches the cells to each other. Gap junctions, totally different category. Gap junctions actually connect cells so that they can talk to each other. A gap junction is like a pipeline that directly connects the cytoplasm of one cell with the cytoplasm of the neighboring cell, so that ions can flow from one cell directly to the other without having to go out to the extracellular fluid and back in again. A direct pipeline. Gap junctions allow for what we call electrical communication. Do you remember we already talked about that? When cells create electricity, that electrical current can go through gap junctions from one cell right into a cell that's connected. So where you see gap junctions, you know cells are talking to each other. And I'm going to give you two important examples and then one by the way. And one of them is epithelial. Ciliated cells are connected by gap junctions. Why does that matter? What does cilia do? Cilia beat. Fluid moves across the surface of the cell due to the beating of fluid. Where did you learn? What was the main location you learned for ciliated epithelium in lab? Respiratory tract, right? Okay, so almost your entire respiratory tract outside of the air sacs is a ciliated epithelium. So how do they know to be in synchrony with each other? How do they do that? It's the gap junctions. As soon as one sends a signal and its little cilia move, then the neighboring one does. So if you've ever seen people do the wave in the stadium, it's kind of like that. They're all synchronized. Okay? You also have gap junctions in some of your muscle tissue, and likewise it coordinates contraction. And then, by the way, there are apparently gap junctions in embryonic brain cells that connect them directly, even though they don't maintain that connection after they're done developing. Okay? So we'll come back as we get to the other tissues and chapters. We'll talk more about gap junctions later. Our fourth kind of junction is a hemidesmosome. And the job of the hemidesmosome is to connect the cells to their basement membrane. So the question is, why is that found only in epithelia? Let's go back to our picture to see where that basement membrane was. Where is it? It's on the basal surface of what tissue? Found only in epithelia. Okay? So why is it called a hemidesmosome? What does hemi mean? Half. So what was a desmosome? A desmosome connects one cell to a neighboring cell. It takes two halves, two cells to make a whole desmosome. If you're connecting to the basement membrane, you don't have another cell as a partner on the other side. So you're just half a desmosome. So why isn't it called an intercellular junction at this point? Why is a hemidesmosome not an intercellular junction? It's not between two cells, right? It's one cell and the basement membrane deep to it. All right, so here's the diagram out of your book, and I'm sorry I didn't put the figure number on this one. Where do you look for your tight junctions? Where are they? Where specifically? Where specifically on the lateral surfaces? You're right so far. You're doing great. So yes, between the cells. It's not off the top. It has to be between adjacent cells. They're at the top. They are close to the apical surface. Okay, why does that matter? Does this cell need to have extracellular fluid? Can extracellular fluid flow through the basement membrane? Fluid? Oh, absolutely. So when you think of basement membrane, think of it like a piece of fabric. Can it get wet? Can it have fluid flowing through it? Yeah. So this basement membrane is basically saturated with extracellular fluid, and that extracellular fluid, I'm going to try to draw as carefully as I can go all the way up even between the desmosomes, so that these cells are literally bathed in fluid. They have to be. Because of the way phospholipid membranes work, they need fluid on both sides. But can this fluid go through and leak out the top? That it cannot do. Okay, so what kind of fluid do I have out here? On the top surface. It's going to be the external environment fluid, whatever that is. If this is the small intestine, then I'm going to have intestinal juice there, which is, again, still mostly a watery fluid. But it's not internal body fluid. This down here at the base, all of the fluid down here is tissue fluid. But the fluid out here out the top, hey, you're in the external environment if you're above the apical surface. That's a free surface. So whatever that external environment is, that's what fluid is bathing those cells. Do I want this fluid mixing freely with the other fluid? Mixing freely? That's the whole job of the epithelium, is to keep them separate and to keep them in the proper composition. So the job of the tight junctions up here, parked up here at the top, is to seal like little Ziploc bags and prevent that fluid from sneaking through. Everybody okay with that idea? By the way, there are some things that can open those tight junctions. Your white blood cells, the neutrophils, the main kind of white blood cell, can actually open them, slither through, and close the door behind them, which is a pretty cool trick. Unfortunately, there are some bacteria that can do it the same, only in the opposite direction. Salmonella is one. Salmonella is a little microorganism that can get right through those tight junctions, open them up, and sneak through to the other side. Okay, questions about tight junctions? Desmosomes look like little stitches. They have fibrous proteins attached to what look like little buttons. And again, they're on the lateral surfaces of the cells. So if that's a desmosome, what's going to be down here at the bottom? That's my hemidesmosome, right? It's like a little button that stitches it to the basement membrane. Okay, questions about cell junctions? Could you identify them from a picture? Okay, we're going to stop and see what we can do with these three questions. Our first one has to do with something we talked about earlier today, but all three of them do reference what we've already done today. So you've got three questions. I'm going to give you at least 5 minutes and walk around and help. Okay, so cancers of epithelia are called carcinomas. Have you heard of that before? Can you name a carcinoma, a specific carcinoma? Anybody? Basal cell carcinoma, typical in skin. The other is squamous cell carcinoma, also found in skin. Okay, and that's the ones most people have heard of. So why would a cancer of an epithelium be common? Think back to what I said earlier today about epithelia in general, but also think about what you learned last week about what kinds of tissues commonly have cancer. So what are you thinking?
They're closer to the surface, closer to radiation.
Okay, so not all epithelia are close to the surface, but for those that are, like the skin, yeah, that's going to be part of it, but that's only about maybe half of it. What's the other half? Okay, so DNA is most susceptible to mutation when cells replicate. How often do epithelial cells replicate? Highly regenerative. Which also, you remember from last week, means they're more susceptible to the side effects of a chemotherapy that hits rapidly regenerating tissues, including hair falling out and nausea because your intestinal lining is epithelial. Okay, make sense to everybody? Questions about that? Number two, why would columnar be better than cuboidal if your goal is protection? Simple answer here. Not surface area. It's not a surface. Well, okay, that's kind of misleading. My statement is misleading. Is there more surface area on the surface, on the free surface? No. It's a matter of how thick they are, right? So the surface area could be entirely the same, the free surface, but the surface area between the two cells, definitely more, right? Okay, so if you look at the top surface of an epithelial cell, can you tell how thick it is just by looking at the top? Do they have a different characteristic shape? No, they all look pretty much the same. So what's the big difference between columnar and cuboidal? It's not their apical surface, it's the cell-to-cell thickness. And the more space there is adjoining the cells to each other, the more room for more desmosomes you have, and the cells actually will kind of fold to each other and hug each other more tightly. Okay, so does it make sense to you that you put simple columnar epithelium in a place like the stomach where you're more likely to put it under too much pressure by eating too much, as compared to, say, the ducts of your salivary glands, which the only pressure they're ever under is just the fluid of the saliva passing through? Okay, make sense? All right, and number three, I referred to, but not by name, pemphigus vulgaris. What happens when your desmosomes in the skin don't hold? Those desmosomes are what hold the layers to each other, and so the whole skin can just peel right off. Looks like giant blisters. And so why would that cause infection then? That skin, remember that solid continuous layer of epithelium keeps things on the outside out. and that includes infectious organisms. All right, so that's our basic run-through. Before we move on to connective tissues, let's back up to our kinds. And let's just have a conversation about where we find the different kinds and what that means. So on the far left, you see simple squamous. And then to its immediate right, simple cuboidal, and then simple columnar. And then what's that on the far right? Stratified squamous. And which one is missing in this picture that you were assigned to learn in lab? Ciliated pseudostratified is not shown here. Okay, so I kind of had to cheat on these pictures. I just used a very simple photo editing software and enlarged them. But the reason I did that was to try to make them look to scale the way they would really look in real life, and that's really hard to do in this particular case, just because of the pictures I started with. But you looked at them in lab, and you know that simple squamous is so thin it just looks like a single line. It's so thin under a light microscope. It's really hard to see how there's anything much there at all. So basically, what you have is two membranes, an outer apical surface and a basal surface, with a very, very thin film of cytoplasm in between. So obviously that's great when you're trying to get stuff moving through. So in the air sacs of the lungs, or in the capsules or the corpuscles of the kidneys, where you're just filtering fluid to form urine, the thinner the layer, the faster the stuff moves. But we also find simple squamous epithelium lining our serous membranes. Y'all remember learning the serous membranes, right? So where are they? Just as a quick review for those that haven't taken the test yet. Where are your serous membranes? Around each lung, right and left pleural membranes, and then around the heart, the pericardial, and then below the diaphragm we have the peritoneal membrane. Each one has two layers, visceral and parietal. All of those are simple squamous epithelium over a very, very thin, loose connective tissue, an areolar connective tissue, very thin membrane. Do these membranes need to absorb anything? Is their job to absorb nutrients or oxygen? No, they're lining internal body cavities that aren't open to the external environment. So why are they there? What are they doing for you? What are those serous membranes doing? They cover the free surfaces of the cavities, peritoneal cavity, pleural cavities, pericardial cavity. Are there any moving parts in any of those locations? Do your lungs move? I mean, they're not moving, but they are inflating and deflating. What about your heart? It's sitting there beating, contracting and relaxing. What about your guts? Oh yeah, they move. They do some moving. So it reduces friction, and how would it reduce friction? How do those membranes reduce friction? We secrete a fluid. We just call it a serous fluid in general, but you can call it pleural fluid, pericardial fluid, peritoneal fluid. The amount of fluid you secrete is tiny. It's supposed to be anyway. It's supposed to be a very small amount of fluid, and that fluid just wets the surfaces so they slide instead of grinding against each other. Any of you ever had pleurisy or pleuritis? Pleurisy is an inflammation of the lining of the lung. And you see that sometimes in cases of people who have some kind of respiratory ailment that just keeps them coughing for weeks at a time, very often with a condition we call walking pneumonia. We call it walking pneumonia because it doesn't kill you that fast. So you're still walking around with your pneumonia. It's a very mild form of pneumonia. A lot of people walk around with pneumonia and don't even know they have it. They know they have a cough. They know they run a fever at night. They don't know they have pneumonia. Eventually, and usually not that long, they'll develop chest pains from coughing. And before you know it, every breath is like dragging sticking a knife in them. That's pleurisy or pleuritis. And that just shows you how important that lubricant effect of the serous membranes is, because that's what you lose, and those membranes start yanking on each other, gluing themselves to each other, and it hurts like the dickens to try to breathe when you have pleurisy. Okay, so the lubricating effect of serous membranes. How thick does the membrane need to be? It's, again, a very thin membrane. So you don't have a lot of friction there ordinarily, so it doesn't need to be a thick epithelium. And the first rule of building in general is don't make it any thicker than it needs to be. Don't waste resources on building something any thicker than the needs of the tissue. So you always put the thinnest layer you can get away with, and in the case of serous membranes, you can get away with simple squamous. That's all you need. Likewise, the lining of blood vessels is simple squamous epithelium. You don't want too thick of a layer because all that would do would be to block the lumen of the blood vessel. And you protect from friction by having the way the fluid flows. The blood flows very smoothly, usually. Okay? So we have these locations of epithelial tissues that are, to a histologist, quite distinct. And the original histologist that studied the microscopic anatomy of tissues came up with not one name, epithelium, but three names: epithelium, mesothelium, and endothelium. Sound familiar? Sounds like mesoderm and ectoderm? Yeah, but look back in your notes. Where does epithelium come from? We had that one slide where I had the germ layers for all of them. Epithelium comes from all three germ layers, okay? So you have to kind of separate, I point this out so you don't get them confused. You have to separate the different kinds of thelia from the different kinds of dermal layers. So you have ectoderm, mesoderm, endoderm. You have epithelium, mesothelium, and endothelium, and there's no correlation between them. All epithelial tissues come from different germ layers, so it helps you a lot to just kind of latch down on where they are. All right, so mesothelium is specifically those membranes lining the serous cavities. Okay, so we talked about pleuropericardium, peritoneum. Those are the mesothelial membranes. Okay? So if I told you that my husband's uncle died of mesothelioma, what did he have? It's not that uncommon, actually. Mesothelioma, what is it? It's a cancer, tumor, of the mesothelial membranes. And it's commonly associated with either smoking or, more likely, working with asbestos or fiberglass. So if you watch a lot of, I don't want to denigrate it, but cable TV, that's what we used to say. Oh, he watches too much cable TV. It's kind of an old-fashioned way of looking at it. But anyway, you see the lawyer's ads, hire me for your lawsuit, those kinds of ads, you'll see mesothelioma being advertised. And it's usually for people that work with asbestos or fiberglass because they inhale those little fibers. Those fibers are indigestible. They work their way through your lungs and into the serous membrane. So when someone has a mesothelioma, typically what they have is a tumor of the pleural membrane, and it's hypersecreting fluid. And there's so much fluid it can cause the lung to collapse. And so what they have to do is insert a cannula, a tube, to just basically drain that fluid off. And in terms of chances of successful treatment, I don't know what they are today. The only people I ever knew that had mesothelioma died of it, so I don't pretend to be an expert on that, though. Okay, so where is the mesothelium? What specifically is it? I just gave you the location. Where do you find mesothelium? The serous membrane. So it is, the mesothelium is the serous membrane's epithelium. Specifically, it's that simple squamous epithelium associated with those serous membranes. Is it an epithelium? Today, yeah. Originally, when histologists first described it, they considered it something separate. Today, we lump it in with epithelium. Okay, what about endothelium then? You might want to conflate it with endodermis, and you'd be wrong. So where is the endodermis? That's the lining of the gut tube and the respiratory tract. That is endodermal origin, but endothelium has nothing to do with any of that. Endothelium is the epithelium that lines your cardiovascular system. It's a simple squamous epithelium. And it lines your blood vessels and your lymphatic vessels. And where it lines the heart, it's the same stuff, but we use a different name. We call it the endocardium. But it's still the same simple squamous epithelium. Why do you want a simple squamous epithelium lining your vessels? Is there a lot of diffusion going on in your arteries and veins? Arteries and veins, no. Capillaries, yes, but not arteries and veins. So again, we just need a smooth, frictionless surface over which the blood can flow. And we want a thin layer because we don't need a thick layer. We don't want to occlude the vessel, okay? So what is endothelium? Specifically, what is it? It's the lining of the cardiovascular system and lymphatic system. What kind of tissue is it? Simple squamous epithelium, okay? So you now know epithelium as an all-encompassing thing. Mesothelium specifically is the simple squamous epithelium of serous membranes. Endothelium specifically as the simple squamous epithelium lining the cardiovascular system. Got it? Questions? It's just a matter of committing that to memory. Okay, moving back to our idea of complementarity. Where would I put simple cuboidal epithelium? Think back to what you learned in lab. Where do you find simple cuboidal epithelium? You have lab quiz on this tomorrow, right?
I guess
Kidneys and small ducts, right on. So kidneys and small ducts. Small ducts, not large ducts. So what's the difference between a small duct and a large duct? The larger it is, the more fluid it carries under higher pressure. The more pressure, the stronger the wall needs to be. So for large ducts, generally, I put a stratified cuboidal epithelium. So what's the difference between a simple cuboidal and a stratified cuboidal? How thick the wall is. The thicker the wall, the more protection it is. Make sense to everybody? So I always use the thinnest layer I can get away with. And for small ducts, simple cuboidal is all I need. Okay? In the kidney specifically, we're doing some transport. And when I'm doing transport, I want a thin layer, the thinnest I can get away with. Why am I not using simple squamous for these kidney ducts? Can't get away with it. Too thin. So the fluid being carried in the ducts of the kidneys has to be under enough pressure that if it were any thinner, it would rupture the ducts. So everybody see just sort of that trade-off between I want a thin layer for transport, but if it's too thin, it's too fragile. Okay? So where do I find simple columnar epithelium? Where do you remember from lab that you find simple columnar? Gastrointestinal for the most part. So the lining of, starting at the stomach and going through the intestine. So what about above the stomach? Esophagus, throat, mouth. What kind of lining do you want there? Stratified squamous, which sounds like it wouldn't be as thick as stratified cuboidal, right? In fact, stratified squamous, as you can see in our image, has lots of layers. So stratified squamous is, in fact, the thickest kind. If it's a stratified cuboidal, it might at most be 3 to 5 layers. Stratified squamous can be 30 layers. And when you look at that picture on the far right, look at the deepest cells. Don't they look cuboidal to you? So why do we call this a stratified squamous epithelium if the cells at the bottom look cuboidal? It's the cells at the top that cause us to identify it as a squamous. So the deeper layers are cuboidal, whether it's stratified squamous, stratified cuboidal, or stratified columnar, it's the cells at the top that cause us to give it the names that we do. And stratified squamous is the thickest, even though it includes at the top the thinnest kinds of cells. Is that enough to be confusing to everybody? Okay, so where do you find stratified squamous epithelium? Lining of friction-heavy environments, mouth, esophagus, pharynx, covering of the skin, vaginal canal, anal canal, those areas that open directly to the external environment and potentially suffer more friction and environmental threat. Make sense? Okay, for the one that's not here, I will attempt to draw a picture on the board. And I'm going to start with the basement membrane. And what I want to tell you is that in a pseudostratified epithelium, all cells attach to the basement membrane. In a stratified epithelium, some don't. Some attach only to the cell below them. But in a pseudostratified, all of the cells attach to the basement membrane. They just don't all reach the apical surface. So I have some cells that are pretty tall and some that are pretty short. So you see that they're all attached at the bottom, but not all attached at the top. Okay? So can you tell from that we have really two different categories of cells here? We have cells that go the whole way, the whole distance, in a ciliated epithelium. Some of these will have cilia. Some of them may be goblet cells, which I know you learned about in lab. Those are typical of columnar cells. Goblet cells are. You don't see goblets in anything except a columnar. You can see cilia on cuboidal or columnar. But in the pseudostratified, it's always columnar. Are you with me on that idea? So what do you want to call these little short guys that don't reach the top? They attach at the bottom. What's the bottom surface called? So we call these basal cells.
There's another form of cancer too, like basal cell.
In a basal cell carcinoma of the skin, it's not going to look like that. It's not a pseudostratified epithelium. But the idea of a basal cell is it's always in the deepest layers. And the cells in the deepest layers that are attached to the basement membrane are always closest to the blood supply. Where is your blood supply in this image? Where's it going to be? Is it going to be here? Can't be here, right? That's the free surface. Is it here or here? It has to be down here. Why? That tissue is avascular, right? The epithelium protects the blood vessel. So the blood vessel can't be in the epithelium. It has to be deep to it. Make sense? But if you're looking at who gets most of the nutrients from the blood supply, the bottom layer clearly gets the best, right? We'll explore that further tomorrow. When we study skin, we'll be looking at stratified epithelia. But for now, we're looking at pseudostratified. Those basal cells have not differentiated yet. The big tall cells have. They're either a ciliated cell or they're a goblet cell. They're specialized. They're differentiated. And they're on their way out. Clock's ticking on their lifespan. How long do they live? 3 to 4 weeks. The basal cells are specialized to regenerate. You could think of them as being stem cells. I don't think we call them stem cells, but you can kind of put them in that category. If this cell goes through the cell cycle and divides into 2 cells, one of the 2 cells will remain a basal cell and go back through the cell cycle again. So it will go immediately into a new G1 phase to redivide. But the other cell, the other descendant cell, will kind of push sideways and grow up into probably a ciliated cell, possibly a goblet cell. So it's going to go G0. Do you remember what that means? It's not going to divide again. It's going to specialize. Okay? So in cases of a carcinoma of the lungs, usually associated with repeated environmental exposure to smoke, like in cigarette smoke, or environmental hazards that you're inhaling on the job, occupational problems like fiberglass or whatever, which cells are you worried about the most? The cilia, actually the ciliated cells respond to some of the chemicals in smoke by losing their cilia. I don't know if they actually lose the cilia or if they just live such a short lifespan that they never actually grow cilia. I do know that in smokers, the ciliation is reduced. Their ability to remove mucus out of the lungs is reduced, and that increases their risk of respiratory infection and other obstructive problems. But it's the basal cells we're worried about. Why are we not worried about these guys? They're already specialized. How long are they going to hang around? Another 3 weeks and they're out of there. And when I grow new cells, do I grow them from these specialized guys? Not grown from the basal cells. So if I have a mutagen, a carcinogen in the smoke that I'm inhaling, I don't care if I mutate the genes in my specialized cells so much, because they're not going to reproduce. But in my basal cells, I do. Make sense to everybody? And so you need to be aware of the differences in a pseudostratified epithelium, that there are at least 2 and potentially 3 different kinds of specialized cells. You always have to have basal cells. Otherwise, it's not a pseudostratified epithelium. And you usually find these in parts of the body, like the respiratory tract, where you are speeding up regeneration. And you're speeding it up because you're exposing it to lots of environmental hazards, potentially. I mean, you may eat, well, if you're like a lot of people, you eat all day. Okay, but you still breathe more than you eat, right? You take 12 to 20 breaths per minute. I don't think you eat quite that much. So which tract is more exposed to the environment, respiratory or digestive? Definitely respiratory. Okay, so it makes sense that it's a pseudostratified epithelium. Actually, that's the only pseudostratified location we assign. It also happens to be the only ciliated location that we assign. And so testing strategy in lab, if you see cilia, and it's on your lab test, you know it's a pseudostratified epithelium, but I want you to know that you can have a ciliated epithelium that's not pseudostratified. For example, the fallopian tubes, which carry the eggs from the ovary to the uterus, are a simple columnar epithelium that is ciliated, okay? Likewise, in the male reproductive tract, there's cilia there. And we learned last week that ciliated epithelia are affected in people with cystic fibrosis. Do y'all remember that? So what happens in CF is they can't secrete the fluids very well, and the mucus tends to bog down in the respiratory tract. In the reproductive tract, male or female, eggs and sperm are not carried properly with the lack of ciliation. So people with cystic fibrosis, first of all, are urged, or counseled, I should say, not to reproduce. Why? Why would you tell a girl with cystic fibrosis that she probably shouldn't have children even if she could? Why would you tell her that? She's going to pass that gene on. Plus, it's going to potentially threaten her own health, never mind the health of the baby. But even if she's dead set on having a baby, she may not be very fertile because the eggs don't move very well. Same with guys, okay? All right, so I think we've kind of gone through all of these. Does anybody have any questions about them? We'll come back to the stratified squamous epithelium tomorrow because we're going to do skin tomorrow, okay? For now, we need to shift gears and talk about connective tissues. You only learned some of the kinds of connective tissue in lab. You didn't learn them all. So for now, you need to know that there are 4 kinds of connective tissue. And the terminology we use kind of varies. That first group, I call it fibrous because that's a good, simple name for you to remember. That's not its actual name. It's just called connective tissue proper. I think fibrous is a catchier name. So what fibrous connective tissues did you learn in lab? If you're saying cartilage, is it a fibrous connective tissue? Look at your second kind of connective tissue. So you learn, let's just go through what you did learn. Did you learn loose connective tissue or real connective tissue? That's a fibrous kind. Did you learn adipose? That's actually a fibrous kind. It kind of gets a little asterisk by it. It's very specialized, but it's still classified as a fibrous. And what about dense? Yeah, that's a fibrous. So in fact, for fibrous, we actually have 2 categories, loose and dense. And we're going to go through those. And then for cartilage, what did you learn? You learned hyaline cartilage, fibrocartilage, elastic cartilage. And that's all there are. There are just those 3 kinds. So you learned them all. The other 2 kinds of connective tissue are bone and blood. Bone is properly called osseous, and blood is called vascular. Those are the proper names for those tissue types. When do you want to learn about bone tissue? How about when we do the bones, which will be Wednesday of this week? Okay, in the long semester it'd be 2 weeks from now, but this is summer school. Okay, and what about blood? When are we going to study blood? A&P 2. So for now, you just don't have to know that much about them. You do need to know that they are 2 of the 4 major kinds of connective tissue, and well, I'll sneak a few more things in there, but not much. All right, so there are some characteristics of connective tissues that almost all connective tissues have in common. And just like I said earlier, you can't always pigeonhole things into a category very neatly. Connective tissue is going to be real problematic for that. So when we talk about the matrix is the stuff around the cells. It is extracellular material. And the matrix has 2 basic components. In a connective tissue, there are almost always some kind of fibers. And then there's stuff that fills in the spaces around the fibers, and we call that stuff the ground substance. Depending on the tissue type, that ground substance can be like water. It can be like molasses, kind of thick and syrupy. It can be rubbery. It can be very hard, or it can be entirely liquid. So depending on the tissue type, the ground substance, which fills up most of that space, can be completely liquid or completely solid. The fibers fall into 3 main groups. And 2 of these you learned in lab already. You learned collagen and elastic, right? So the characteristics of these fibers are very helpful for you to know. Collagen fibers, to me, are like good solid ropes. What is that? Is it outside the room?
I guess.
It must be the weed eater out there or something. Yeah, I can hear it from here, so very weird. Okay, collagen, tough, flexible. What can you do with a rope? You can bend it, right? You can tie it in knots. You don't usually do that in your tissues. Can you stretch it? Is it very stretchy? Not very. If I want something to stretch, what am I going to put in there instead of collagen? That's where I'll put my elastic fibers, which, like rubber, are going to stretch and then recoil. Okay, and it's the recoil that makes it elastic. When things lose their elasticity, they lose the ability to snap back. Okay, so they may or may not lose the ability to stretch, but they definitely lose that springy recoil. For example, an old worn out pair of socks. It's lost its elasticity. Tell me about them. They fall down, they sag, they stretch out, and they won't snap back. Tell me about skin that's lost its elasticity. Okay, sags and wrinkles. What about lungs in someone with emphysema? All stretched out, no nice springy recoil. Okay, so elasticity is the recoil, the ability to spring back after it's been stretched. Okay? Our third fiber type is called a reticular fiber. Reticular means network. So when you see rete or reticular, think of kind of a mesh or a network of fibers. And in this case, they're very thin and delicate, but they are made out of the same protein that makes our collagen fibers. That is the protein collagen. There are different forms of collagen, and different tissues make different molecular varieties of collagen. And so I'm just going to have to tell the guys not to. It is distracting. It is the weed eater. Yeah, so they never weed eat back there. So after they do it today, they won't be back until this time next year, I'm pretty sure. But I'll tell the maintenance guys, to do that. All right, so here's just a little bit of chemistry. I'm going to try not to overdo it, but I think it's important for you to review your chemistry and to relate it to things that might make some sense to you. So we're going to start with something called a glycoprotein. Can you tell me what it's made of? What are the 2 parts of a glycoprotein? What's the obvious part? Protein. What's the glyco tell you? Carbohydrates, sugar groups. Okay, so looking at that thing that looks kind of like a bottle brush, a test tube brush, The core of it is a protein, and all the little bristles of it are your carbohydrate groups. And are they polar or non-polar? Carbs? Polar, 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 polar. Lots of OH groups on carbs. Okay, so it just sucks up the water. Just sucks it right up. All that gray you see in the background, that's all the water. And in fact, that water is tightly linked in between all the little bristles of that brush. There are a couple of named molecules there. I don't test you on them, but I do like to introduce them to you. One is chondroitin sulfate. Have you heard of that? Do any of you take supplements for your joints? All you guys are pretty much in the I'm too young for that category. But for us oldsters, or if you have an old dog, you might give them chondroitin sulfate. It's just one of the nutritional supplements that people take and hope that it's going to help them with their joints. And this is where it's used. It's used to make these glycoproteins. These glycoproteins, depending on how much or little of them you put in water, will, if you put too much, will turn it completely solid. So the best example I can give you here is like Jell-O. And unfortunately, these days, very few of my students have ever made Jell-O. So I'm still in the habit of asking, how many of you made Jell-O? Wow, that's a pretty high. I'm going to tell you, usually I have one or two that are willing to admit that they've made Jell-O. So how do you make Jell-O? Add water to what? The little package. What's in that package? Is it a powder, a syrup? What is it? It's a powder. It's this dry powder. So tell me I can take this small handful of dry powder and throw it into 2 cups of water and make the whole thing solid. That's magic. I'm sorry. It's insane. So what's going on? They're like little sponges and they suck up all the water. Have you ever made Jell-O jigglers? Any of you? How do you make them? I only did them once with my kids. The first thing they did was throw them at the wall to see if they would stick. That was a one-time thing. So how do you make Jell-O jigglers? What do? You don't need any other ingredients, do you? What do? Less water. So you have a greater powder to water concentration, and it makes it more like gummy bears. Okay, so that's the difference. How much of this stuff, this gelatin stuff, compared to how much water, that's going to determine the consistency of my Jell-O. If I leave it in the fridge too long and forget to cover it, what's going to happen to it? It's going to lose water by evaporation and just turn into this little hard rubbery thing. Well, that's like cartilage. So cartilage has this in its matrix. Your loose connective tissues have quite a bit of it in their matrix. Your dense connective tissues have more fibers, so not as much room, but they still have this stuff. If it's a fluid or gel-like consistency, or like cartilage, it's just pretty solid but rubbery solid, then it's using this glycoprotein stuff. Part protein, part carbohydrate, just absorbs water, just absorbs it like crazy. Okay, so that's the way that works. So tell me how much water there is in cartilage. If there's 2 cups of water in 2 cups of Jell-O, how much cartilage is there, or how much water is there in a hunk of cartilage? Just like Jell-O, it's mostly water. Okay, so living cartilage, even though you're used to seeing it either dissected or more likely in chicken, where it feels pretty solid, the gristle at the ends of bones, that's hyaline cartilage. It's solid but rubbery. You don't think of it as having a lot of water in it. It's full of water. And in the living body, as you put pressure on the cartilage, you squash the water out of it. And then when you let the pressure off, more water flows into it. Okay? So it's important for you to have some idea of the consistency of that brown substance. All right, let's get into our categories. We have loose and we have dense. We have 3 kinds of loose connective tissue, areolar, reticular, and adipose. Can you tell me what kind of fiber is going to be found in a reticular connective tissue? What kind of fiber? Just guess. We'll go back to our fiber types to help you out. You see your 3 kinds of fibers listed at the bottom of the screen. It would be the reticular fiber. So when you see a reticular connective tissue, guess what? It's got reticular fibers. Remember, they're small and sparse and delicate and thin. And so when you look for where reticular tissue is located, what you find is the fiber's not the most important thing. The cell is more important. Okay, 4-hour loose. Why is adipose listed there? What is adipose connective tissue full of? Yes, this is a trick question. It's full of adipocytes. If you extracted all the adipocytes from it, guess what's left? Basically a loose connective tissue. So in embryonic origin, all the places where you have adipose tissue, whether it's your superficial fascia or behind your eyeballs or around your heart and kidneys, everywhere you find collections of adipocytes in the embryo started out as just plain areolar loose connective tissue. And then it got invaded by adipocytes. So that makes it classified as a connective tissue even though there's not much matrix and it doesn't really connect anything. Okay? It has to do with embryonic origin. On our dense guys, we need to focus on the differences, and you'll notice here that of the 4 kinds listed, you only learned one of those in lab. You learned regular, collagenous, dense connective tissue. Okay? You have to know all 4 of them for lecture, which means you have to know why they are the way they are, and you have to know the locations of them. So we're going to go through these differences between regular and irregular, and you're already somewhat familiar with the differences between collagen fibers and elastic fibers, so I think the rest of it will make sense. So if I have a regular connective tissue, this is the one you saw in lab, so tell me what you know about the fibers in a regular dense connective tissue. What do they look
They
like?
have a pattern.
Okay, what kind of a pattern?
They go the same way.
They all go the same way. And in the one you saw in lab, they tended to be kind of wavy, right? And that was collagen. Okay? If it's elastic, it looks more like this. Okay? If it's irregular, that's regular. If it's irregular, what you find is bundles of fibers that run in different directions. Not all the fibers running in the same direction. So a regular connective tissue has the fibers parallel to each other in arrangement. They all run the same direction. And irregular, they run in all directions. That's a pretty clear difference, don't you think? Even though it's not that easy to draw. And when you look at the images of these in your textbook, let me see if I have those in here. Yeah, I do have some. Look at the middle figure up on the screen, and you can see that you're just seeing little bits and pieces because you're slicing through them. So it's just not that pretty. Looking at the top figure, doesn't that look familiar from lab? And then looking at the bottom figure, can you tell the fibers are all running pretty much in the same direction? Okay. But squiggly looking? Yeah. Okay, for lecture purposes, I will not do visual identification. That's what you do in lab. So you look at the pictures in lab, just of the ones assigned in lab. But for lecture, you need to know them all and you need to understand why they are the way they are. So if I pull, this could be a tendon, regular, dense, collagenous. It could be a tendon. If I pull on it in the direction that the muscle is attached, it's always going to pull this way, right? Along the alignment of the fibers. What would happen if I accidentally pulled it the wrong way? It would shred the fibers apart from each other. Okay? So whereas with the irregular, it doesn't matter which way I'm likely to pull them, I'm going to have some fibers aligned in that direction. So does it make sense to you that you find regular collagenous in tendons and ligaments? What do tendons and ligaments do? What do tendons do? I have a very specific thing. They connect muscle to bone. Your muscles always contract only in one direction. Your tendons always get pulled the same way. Okay? Ligaments attach bone to bone. Likewise, they tend to, because of the way the joints are arranged, they tend to pull only in one direction. But you also have fibrous capsules around your joints. Those are irregular, collagenous, dense connective tissue. And you can rip those suckers. And that, first of all, it's very painful. And second of all, it's hard to fix. So if you've ever had a rotator cuff injury, then you know that once you pull those collagen fibers, they tend not to go back. These tissue types are poorly vascularized. What does that mean? Epithelium was avascular, no blood vessels. These guys, poorly vascularized. Very poor blood supply, very few blood vessels. They have to withstand a lot of pressure. Blood vessels are not that strong. So if you put too many blood vessels in here, it weakens the tissue and just makes the blood vessels more likely to rupture. Okay, questions about that? All right, so how well do they heal if they're poorly vascularized? Not very well. And once you yank those collagen fibers, yeah, there's no way to push them back where they came from. So it's really kind of hard to do that. Okay, so dense collagenous tendons and ligaments. What about dense irregular? This stuff's really good at puncture resisting. You can't say puncture proof. Things can get through. Dog bites, for example. I happen to know. But they're puncture resistant. Collagen fibers are tough, and as long as they run every which way, they put up a lot of resistance to anything trying to push their way through. Can't be twisted easily. So the pericardial sac, that fibrous sac around your heart, puncture-resistant protection. Okay, you have capsules around your kidneys. You have the whole white of your eyeball, this kind of tissue, puncture-resistant. The dermis of your skin, puncture-resistant. Okay, collagen is tough stuff. It's strong, but it doesn't stretch very well. So if I want stuff to stretch, I need to throw some elastic fibers in. The dermis of your skin is actually a mix. It's another one that doesn't fit neatly into a category. Mostly collagen, but enough elastic fiber that, for example, the skin across your knuckles can stretch without sagging. So what happens when people have burns across a joint? It doesn't stretch anymore. What grows back as scar tissue is collagen without the elastic fibers. So if you don't do proper therapy with them, the skin grows back so tight they can no longer flex their fingers. With proper therapy, though, when they extend the fingers fully, the skin kind of gaps up because it has lost its recoil. Okay, so we appreciate the elasticity in the dermis of our skin. It's what I would call a mixed, dense connective tissue, irregular, mostly collagenous, but with enough elastic fibers in there to keep your skin recoiling until you get old and saggy. Okay. What about our elastic tissues? Some books don't make a distinction. Yours doesn't make a distinction between regular and irregular. I'm going to ask you to make that distinction because we see it in different locations of the body. So in your vocal cords, what you got? The lining or the interior of the vocal cord is a dense connective tissue. Do I always stretch them in the same direction, my vocal cords? Like a guitar string, yeah. Okay? Put it under the same tension in one direction only. So I want to regularly arrange tissue, but I need elastic fibers so I can stretch them. Make sense? Vocal cords, regular elastic. Irregular elastic, where I need to be able to expand in all directions. How about your lungs? How about your arteries? What happens to arteries when they lose their elasticity? They can't expand anymore. And your blood pressure goes up, and then they harden. You get arteriosclerosis. Okay? So that's just a quick review of our dense connective tissues. All right, so I mentioned this already, that dense connective tissue is very poorly vascularized and doesn't heal well. A realer tissue, that's your loose connective tissue. What locations did you learn from lab for that? Locations of a realer connective tissue, the loose connective tissue. Where in the blood vessels? Not the lining, that's endothelium. It's got muscle next, smooth muscle. It's the outer layer, particularly the largest blood vessels. But I think the most important location to learn for your loose connective tissue or areolar tissue is under almost any epithelium anywhere. If you cut through an epithelium, if you cut through your skin, you got that paper cut and it did bleed, what did you cut into? This guy, loose connective. It's under any epithelium anywhere with minor exceptions, which eventually we'll get to probably in AMP2. Okay? Epithelial tissues, we already know, are always avascular, right? So where's the blood supply for any epithelium? Right up under it. What tissue's right up under it? Areolar. So where are all the blood vessels for your epithelium? In that areolar tissue. Very, very good blood supply. What about adipose? Great blood supply, no blood supply, somewhere in between. Take a stab. What about the metabolism of your adipose tissues? As high as muscle or not? Which one has a higher metabolic rate, muscle or adipose? Muscle, definitely. You've got that scope for activity in muscle tissue. So the more adipose tissue your body is composed of, your body composition of adipose, the more of it's adipose, the lower your resting metabolic rate per unit of weight. Okay, adipose, not that fast of a metabolism, but it does have a blood supply. So I would say it's kind of in between. It's got a good blood supply, but it's not a great blood supply. It's not avascular. Okay, we're going to go fairly quickly through the cartilage, and you'll see a lot of its kind of review. Let's start at the bottom. All cartilage is avascular. What does that mean? Does not have a blood supply. Why not? Why don't you want to put a blood supply in cartilage? What did I just tell you? It's tough, right? I'm going to squish it a lot. I'm going to put it under a lot of pressure. And like my dense fibrous connective tissues, you know, you could be tearing blood vessels. So cartilage is avascular. What does that tell you about how thick it can be? Can I put really thick cartilage anywhere? I mean, some of my bones are pretty big around, but I can't make cartilage that thick. An avascular tissue relies on diffusion from the outside to get nutrients to the inside. The thicker it is, the further the middle is from where the nutrients are diffusing from. And we already have, I would say, probably the biggest thing in that regard that we have to worry about is the lens of the eye. Does it make sense to you that your lens is avascular? Don't you have to see through it? What would happen if it had blood vessels in it? They'd be in your way. You'd be seeing blood vessels. So the lens is avascular. It's crystal clear. It's made of cells, living cells, that maintain these crystalline proteins. And as those cells live out their lifespan and die, the proteins that they've been maintaining start to just sort of degrade over time, and your lens gets cloudy. What do we call a cloudy lens? That's a cataract. And so what we see is with the development of cataracts, usually it's age-related, and it's because of a reduction in the ability of the lens to get those nutrients diffusing into it. So how big is the lens of your eye? The thickness of it? Yeah, maybe half a centimeter, I'm thinking. Not very big. So likewise, cartilage is never going to get very thick. It just doesn't because it's avascular and it depends on diffusion of nutrients in and out. All right, and because of that, because it's avascular, how well does it heal? Not too good. So if you tear a cartilage in your knee, I've had students tell me, I had torn cartilage, and my doc said, let's just watch it and see what happens. And I'm like, it's not gonna heal. Torn cartilage doesn't heal. So generally, if you have torn cartilage in the knee, there's this little pad of cartilage. There's actually 2 of them in each knee. And when it tears, it just gets a rough edge. It's like it splits and it gets a rough edge. And then every time you move your knee, it's rubbing against those membranes in the joint, and it hurts like anything. Plus, it's not very stable. So what they'll do when they do arthroscopic surgeries, they just go shave the rough parts off and hope that fixes it. If it's too unstable, they'll actually have to go in and try to repair. But it's not a good scenario all the way around because it doesn't heal. Okay. You learned 3 kinds, hyaline, elastic, and fibrocartilage. What kind of fibers are in fibrocartilage? What's the fiber type in fibrocartilage? What's the fiber type in elastic cartilage? That's a trick question. That's got to be elastic fiber. So you put elastic cartilage where you need the support of something solid, but you need it to bend, and don't forget the recoil. So can you name locations of elastic cartilage? The epiglottis, right? What's that? It's that little flap in your larynx that folds down and it flips back up when you're through swallowing. The auditory tubes, what do? They drain the fluid from your middle ear cavity to your throat. They open just long enough for the fluid to drain, and then they snap shut so that bacteria in your throat can't crawl up to your ear. Although in children, sometimes that's exactly what they do, and you get a middle ear infection. Okay? Third location, the pinna or auricle of the external ear. Now, why on earth does that need to move? Most of us don't go around flapping our ears. But we're built like other animals. Have you ever seen a cat taking a nap? What do their ears do? If you're making noise, they're moving like little radar dishes. Okay, what about a horse? What do their ears do? They're flicking flies usually. Okay, so that elastic cartilage in the pinna of other animals has functions that are lost in the human. Because my dad could wiggle his ears, but it used to amaze us as kids, and that's about the most use I've ever seen for movement of the pinna. That's about it. All right, so that's elastic. Now back to fibrocartilage. What kind of fiber? It's not elastic. What's left? It's collagen, and what's the reputation of collagen? It's tough, it's strong. So I put fibrocartilage in places where I need a really tough cartilage that can withstand a lot of pressure, and that is in certain kinds of joints. The joint between the hip bones in the front, the pubic connection, the symphysis pubis, and between the vertebrae. I have the cushion between my vertebrae, my intervertebral discs, that the capsule around the outside of it is fibrocartilage. What happens if that ruptures? That's bad. That's a herniated or slipped disc, and that can pinch nerves. So that's not a good thing. So does everybody appreciate where we have fibrocartilage? Plus those discs I was talking about in your knee, yeah, those are fibrocartilage too, really tough. You have them in your knee, you have them in your jaw joint. So if you're in the habit of opening beer bottles with your teeth, you should probably stop that anyway, but you have a nice tough pad of cartilage there, and I believe in your thumb joint as well. So fibrocartilage in joints that undergo more pressure than usual. Every other bit of cartilage in the body, hyaline cartilage. But again, there are places where it's sort of a mix. So in the cartilages of your nose, you're thinking, man, that's got some springiness to it. Yeah, it does. It's actually got some elastic fibers in it, just not enough to be classified as an elastic cartilage, not that springy. So in between and not in a nice, neat category like most of the other ones we've done. Okay, so I'm hoping that those explanations will help you as you're learning the classifications for lab. Okay, we've kind of already done our first two. I went through those with you, right? Anybody have any doubts about those? All right, so what I want you to do in your groups, come up with an answer just for the last one, why cartilage is avascular. Leave your papers on the table. What are we doing tomorrow? What's on our list for tomorrow? I tell you what, I'm probably going to just assign the rest of that chapter. Yeah, no, I'll probably finish this chapter and then we'll do skin tomorrow. If you have not yet taken your lecture test, you have the rest of today. A reminder, those of you that are going to be in CAP, what I'd like, since it's your first CAP, what I'd really prefer that you do No, let me retract that. If you're ready to go take your test from here, it's right next door, go take your test. Don't forget to go to CAP after that, though. And everybody in the afternoon CAP, please check into CAP today. Either go there first, study for a while, then come take your test, or go take your test and then go to CAP. How many of you are planning to go straight to testing from here? Just so we have some idea. Okay, it looks like no waiting then. And then the rest of you, I assume, are going to go to CAP for a while and study. Do you have to have your test done by 5 o'clock today? No. How late have you got? They will let you in as late as 7. What happens if you get over there at 7:01? You're too late. And that's by their clock. And they won't listen to you argue. So don't be late to go take the test. All right? I'll see y'all tomorrow.